Hi everyone, thanks for joining me today. My name is Alissa Ball and I'm an undergraduate here at Michigan State University. Today I'm going to be showing you my experiment on biosorption of heavy metals by fungi. So we start with our problem. In this case, that is heavy metals. Heavy metals are used and produced by tons of different industries, namely mining, but also including electroplating and the dye making industry. They're very difficult to remove because they're not molecules that could be broken down. They're also toxic to both animals and plants, and some are carcinogenic, making even trace amounts harmful. So, how do we currently remove these from our environment? We have two main methods, those being chemical precipitation, which uses lime to precipitate out the heavy metals, and we also use ion exchange, which requires a synthetic resin to precipitate out the metals. So, why don't both of these processes work well? Well, neither of them can collect trace amounts. They also both leave behind toxic metallic sludge that is still very dangerous and still needs to be treated further. So, with that background information, we can ask some more questions like what is biosorption? So, biosorption is when any type of biological material accumulates contaminants. And we can then ask if fungi use biosorption. And yes, they actually do. We found this out in the 90s. They're able to do this dead and alive. And fungi are actually cheaper than other biological material we have on the market as well. With that, we come to the idea of could fungi maybe be used to clean up heavy metals better than current methods? So, with that in mind, I give you the hypothesis that under optimal conditions, there will be fungal species that can efficiently decrease heavy metal concentrations. So, some goals of such a study would be to obtain data to back up the existing literature on this topic, as well as to obtain data on metals that haven't been looked at yet. I'd also like to explore the reusable capabilities of the fungal biomass, as well as build a much better understanding of the fungal mechanisms being used to uptake these heavy metals. So, we need some materials for this experiment. And with that, we started with fungal acquisition. So, soil was taken from nickel mines in the Upper Peninsula of Michigan. Fungi was then separated from the soil, cultured, and purified. You can see here I've got some diluted soil samples photographed for you to see. After that, we did PCR analysis to identify all the species that were present in the samples. So, in that analysis, we found 13 different species of fungi. On the left, I've got Trichoderma ricci, and on the right, I have four other species that we found. I just wanted to show you how diverse all of these different species are compared to one another. So, all of these species are presumably tolerant to high levels of nickel. So, hopefully, they're also tolerant to high levels of other heavy metals as well. Some other materials that we would need besides those fungal cultures would be malt extract agar, nutrient solutions, heavy metal solutions, as well as a mass spectrometer, an autoclave, and a lot of other miscellaneous lab equipment as well. So now we get on to the plan. So this is just a very basic overview of what the experimental procedure would look like. We would begin by growing fungi on solid agar to purify the sample. We would then grow samples in an optimal growth solution. On the left, you can see me pipetting out a growth solution, and on the right, you can see an orbital shaker being used to stir up the fungi while they're in that solution. After a while, we can harvest the fungal biomass and add that to a heavy metal solution. We allow the biomass to accumulate metals for up to 12 hours, and we measure heavy metal concentrations at different intervals and at the end. Then, of course, we will assess our data, refine anything if needed, and define results. And of course, some things I hope to find out that I can discuss at the end of this experiment would be how effectively can fungi remove heavy metals overall? I also want to know if fungi are more effective than all the other methods we have for heavy metal removal. I also want to know some of the best species specifically for this job, as well as what are some drawbacks, if any, to using fungi. Hopefully, I'll be able to assess whether these results can be applicable to soil, since that is the main target at the very end of this. I should mention that this is one of many future experiments. We can get very complex with this topic, so I definitely hope to be doing this for quite some time. And in the end, as well, I just wanted to give many thanks to Sophie Gabriziak for soil collection, fungal culturing, and support, Dr. Wei Zhang for support and materials, Dr. Greg Benito for equipment and support, as well as the Pathway to Research program here at Michigan State University for mentorship and support as well. So thank you all for taking the time to hear about my experiment. I appreciate it very much.